Good afternoon to all the students. So, today we are going to explore the discoveries part from molecular basis of inheritance. In previous sessions from this chapter, we have clearly learnt on the genetic material, that is DNA, differences between DNA and RNA. We have also learnt the single stranded and double stranded DNA, what are the bonds involved in it, what are the components of a single nucleotide, what are the bonds involved in a single nucleotide, and we have also learnt on salient features of DNA and then Chargaff's rule. Then we have looked into replication part also. But then today I am discussing on the search of genetic material. There was a huge debate on what is the genetic material actually. Is it DNA or RNA or any other cellular component that is being transforming across the generations. From principles of inheritance and variation, Gregor John Mendel has clearly depicted the changes that are coming across the generations, be it phenotypic changes, and from chromosomal theory of inheritance, we have clearly learnt on what is the changes that are happening inside the chromosome adjoining with chromosomal theory of inheritance from cell biology, right. We have learnt that. And in molecular basis of inheritance, what is the genetic material that is actually being transforming? The discoveries have first started with transforming principle. This experiment was done by Frederick Griffith. And the model organism chosen for this experiment is Streptococcus pneumonia. I think you have come across this experiment in your previous classes as well, but then from this experiment there is in continuation with, like, to prove that DNA is the genetic material, in continuation with this experiment we have few other experiments also that we are going to learn now. So, here the model organism, which is Streptococcus pneumonia, two strains of this bacteria are considered. One is S strain and the second strain is R strain, right. S strain of bacteria, here you can see the cell wall of bacteria is made up of what? Mostly it is composed of peptidoglycan, and outer to this it has a smooth shiny coat which is made up of polysaccharide. It has a smooth shiny polysaccharide coat. Which makes the bacteria virulent and it survives in the host immune system. When this S strain Streptococcus pneumonia enters into the host organism, it can survive because it is surrounded by a shiny smooth coat. The bacteria is surrounded by a shiny smooth coat which can survive in the host immune system, and as the bacteria is surviving, it will kill the host, right. So, this S strain is virulent. Whereas R strain bacteria, if you take R strain bacteria has a rough wavy coat over it. It does not have any coat, it just has a rough wavy cell wall, okay, like this. It has a rough wavy cell wall, it does not have any coat over it surrounding it. So, it cannot protect itself in the host immune system, okay. This R strain cannot survive in the host immune system. As such, R strain gets killed inside the host immune system and R strain is non-virulent. Fine, right. So, we have got to know about two strains of Streptococcus pneumonia. One is of S strain and the second is R strain. S strain has a smooth shiny coat over it which is a polysaccharide coat and it is virulent, whereas R strain is non-virulent. It does not have any protective coat over it. Now, if you observe, when living S strain is injected into mice, as it is virulent, it will kill the mice. This is the first experiment he has done. the living S strain, which is virulent strain, when injected into mice, mice dies. When living R strain, second experiment, when living R strain is injected into the mice, mice is alive. Got it? Mice is alive. With that, we can say that S strain is virulent and R is non-virulent. Third experiment, when this S strain is heat killed, the culture, that is S strain Streptococcus pneumonia, when this culture is heated, the cell wall gets disrupted. This we will call it as heat killing. Okay. When S strain is heat killed, such S strain, when it is injected into the mice, mice is alive again, right. But in the fourth experiment, what did he do was, in the fourth experiment, to this dead or heated S strain, the living R strain is added, and this culture is injected into the mice. Okay. To the dead S strain, living R strain is added, and this culture is injected into the mice, mice dies. Mice dies. Got it? Then he has said, he has proved that from the fourth experiment, there is some transforming principle that is being absorbed by the dead R strain. There is some transforming principle that is absorbed by the living R strain from the dead S strain, which made R strain virulent. Okay. There is some transforming principle where R strain has absorbed the S strain, has absorbed the dead S strain, and this made the living R strain virulent. But he did not prove what is the transforming principle, is still a question mark. What is the transforming principle is still a question mark. Okay. So, from this very first experiment, which is done by Frederick Griffith, what is this first experiment? He just said that there is some transforming principle that is being transferred from living S strain to from dead S strain to living R strain. There is some transforming principle there beside it, so that the living R strain became what? Virulent. Something is transferred from dead S strain to living R strain, which made R strain virulent. But what is that transforming principle exactly? Is it DNA? Is it a DNA? Or is it RNA? Or is it any protein? Or any cellular component? Or any cellular component is still a question mark. Is still a question mark. Got it? So, now to prove what is that transforming principle exactly, the experiment is being continued by Oswald Avery, Colin MacLeod, and MC Carty. These three scientists have done biochemical characterization of the transforming principle. What is that actually transforming from dead S strain to living R strain, right? So, that was proved by biochemical characterization. What did they do? To the culture medium, this is the culture medium, right, which has living R strain and dead S strain. Living R strain and dead S strain. To this culture medium, when they have added protease, protease will digest what? Proteins. Remember 3 points: protease digests proteins, DNase digests DNA, RNase, digest RNA, right. So, to this leaving S strain and dead, leaving R strain and dead S strain to this culture medium, when proteinase is added, of course, it will kill proteins, but there is no effect on the transforming principle. There is no effect on the transforming principle. It is causing the same effect. Okay, there is no change that you can see. The mice will be dead here. But second, this is the first phase. Mice dies. There is no effect. It is same, right? Fine. So, to this culture medium, first what added? What is added? Protease is added. When protease is added, from this medium, proteins are being disrupted. Proteins are being disintegrated. But then there is no effect, there is no change, mice dies as usual. So, there is no effect on transforming principle. In the second experiment, when you see, to this culture medium, RNase is added. Okay, to this culture medium, RNase is added. To this culture medium, RNase is added. And here if you can see, again there is no effect on the transforming principle. The mice dies as usual. Mice dies as usual. In the third phase, if you see, to this medium, when DNase is added, it will disintegrate DNA, right? And here the mice is alive. The mice is alive, which means there is an effect on transforming principle. Yes or no? Here, when DNase is added and when DNA got disrupted, in simple terms, in the absence of DNA, there is no transforming principle you can see from dead S strain to R strain. There is nothing which is transformed and the mice is alive. So, with this they proved that DNA is the genetic material. DNA is the genetic material which is being transferred or transformed. DNA is the underlying transforming principle that is being transferred from the dead S strain to living R strain and made R strain virulent. Once again, I am explaining you. So, the transforming principle is first identified by Frederick Griffith on the model organism Streptococcus pneumonia. He said that there is some from the fourth experiment, he said that there is some transforming principle underlying where living S strain has absorbed from the living R strain has absorbed from the dead S strain, which made R strain virulent. This is the transforming principle. He said there is some transforming principle underlying it, but there is a big question mark on what is the transforming principle. Is it DNA, RNA, protein or any cellular component? This is answered by biochemical characterization which is done by three prominent scientists. Those are Oswald Avery, Colin MacLeod and M.C. Carte, wherein to this culture medium which contains living R strain and dead S strain, first come first, protease is added. Protease is added. Protease will denature or disintegrate the proteins. When this happened, you can see the same effect that mice dies, that this means that there is no effect on the transforming principle, even in the absence of proteins. Here in the second phase, when RNase is added, in the absence of RNA also, there is no effect on the transforming principle. But in the third phase, when DNase is added and there is no DNA, in the absence of DNA you can see a clear effect on the transforming principle because the mice is alive. There is nothing that been transferred from dead S strain to living R strain. Here the R strain is non-virulent only. R strain is non-virulent. Fine, right? R strain is non-virulent in the third phase when DNase is added. That means in the absence of DNA, R strain is non-virulent itself. We may get some of the statement based questions like, when proteinase is added to the culture medium which has living S strain and dead R strain, you can see virulent R strain. When RNase is added, again there is no change, you can see the virulent R strain itself. But in the third phase, when DNase is added, you can see our strain is non-virulent. Got it? This is the effect on the transforming principle that we can see, and this experiment is conducted in the year 1944. And the story is not ending here. We have identified that there is some transforming principle from Frederick Griffith's experiment, and second phase biochemical characterization is done, and we have seen that DNA is the genetic material. Till here it is clear, but then the story is not ending here. There should be some standard proof that DNA is the genetic material itself, and this is proved by Hershey and Chase experiment, where he has given the unequivocal proof that DNA is the genetic material. So, in the third phase, this will end the question mark that DNA is a genetic material. Of course, it is answered from biochemical characterization, but then it is not proved, right? There should be the transformation that we can see across the generations, proving that DNA is the genetic material. For that, the unequivocal proof that DNA is the genetic material is proved by Hershey and Chase experiment. What did they do? They have taken T2 bacteriophage. T2 bacteriophage. We know what is bacteriophage, right? Bacteriophage is one which affects the bacteria, which infects the bacteria, right? So, here T2 bacteriophage is taken. T2 bacteriophage. T2 bacteriophage is taken, and we know bacteriophage has a tadpole structure. This we have learnt in NCERT 11 in the previous class itself, where the bacteriophage will have tadpole structure with 4 components in it, like it has a head, inside head there is a genetic material, and head has a neck or a collar, and this has tails likewise, and at the end of the tail fibers you can see lysozyme. So, what are the 4 components that we come across the bacteriophage? One is the protein coat. Protein coat is made up of what? Capsid. Capsid is the protein coat that is made up of amino acids itself. Protein coat, which is made up of, it has capsid, and this consists of amino acids. So, this is protein coat which is capsid. It consists of what, ma? Amino acids, right? So, we have come across many types of amino acids, be it aliphatic amino acids, aromatic amino acids, sulfur containing amino acids like cysteine. Likewise, we have come across many amino acids. It is composed of amino acids, and inside you can see the genetic material. Genetic material which is double stranded circular DNA. Double stranded circular DNA. And this first component is what? Head. Head, collar or neck, collar or neck, and these are tails, and this is tail fibers. Tail fibers at the tip, it has what enzyme? Lysozyme. This lysozyme will disintegrate the wall layer of bacteria. This will disintegrate or disrupt the bacterial cell wall, so that the genetic material can be easily injected into the bacteria. So, these are the components. In the head region you have protein coat which is made up of amino acid. We address this as capsid. It contains all types of amino acid, inside which you have genetic material which has double stranded circular DNA, and then collar or neck, tail fibers. At the end you have lysozyme. This lysozyme will disintegrate or disrupt the bacterial cell wall. Here if you see, we have 3 steps, 3 underlying steps. First, infection. Second step is blending. Third step is centrifugation. Yeah, here we go with this. First, now you have understood the components of a bacteria, right? And inside the genetic material, when I have to say, this is double stranded circular DNA. DNA is a polynucleotide chain, that you know, right? Polynucleotide chain, where the components of a single nucleotide, it consists of a phosphate group, nitrogenous base and a sugar, right? These are the components of a nucleotide. Phosphate group is negatively charged, right? So, in one culture, let me suppose that I have 2 cultures, one culture of bacteriophage in one plate, and in the other plate I have a second culture. In the first culture, I am taking a heavy isotope or a radioactive isotope of phosphorus. That isotope is P32. Okay, consider this as the second and this as first. Okay, to the second culture plate, if you see, to the second culture plate, we have taken bacteriophage, which has an isotope of P32, right? Here this isotope is added where? Into the genetic material. Remember this carefully, okay? Because where the isotope is used in this 2 culture medium will tell you about the entire experiment. Because the single diagram will be there in your NCERT and the type of experiment will be a question mark. When multi-statement based questions are put from this concept, you will face a confusion. So, you should be clear on the experiment, okay, when the diagrams are clearly mentioned. In one culture plate, that is in the second culture plate, what are we doing? We have taken an isotope of phosphate, okay, that is P32, which is integrated into the genetic material. In the first bacteriophage, if you see, first bacteriophage, here this is the culture medium, first bacteriophage, this is the culture medium, wherein the isotope is integrated into the sulfur containing amino acids like cysteine, wherein heavy isotope or radioactive isotope of sulfur is taken, that is S35. S35. Got it? This is integrated where? The first, this is integrated into the protein coat, right? This is integrated into the protein coat and this is integrated into the genetic material. You are clear, right? This is the first culture plate and this is the second culture plate. To the first culture plate, S35 radioactive isotope is being integrated into the protein coat, got it? This is the second culture plate. Here the radioactive isotope is integrated into the genetic material, that is P32, got it? The genetic material is here P32, which is integrated, radioactive isotope is integrated into the genetic material. You are clear on this too, right? Now, the very first phase we are doing is taking these 2 cultures, one with protein integrated radioactive isotope, that is S35, second culture plate with genetic material integrated P32. These 2 are infecting a bacteria, 2 radioactive isotopes, one from genetic material, this from proteins, this is from genetic material radioactive isotope, this from proteins, is integrated or infected into the bacteria. Once it is infected into the bacterial culture, the entire culture is blended, okay. It is blended in the second step. What is blending? The genetic material of bacteriophage will get integrated into the genetic material of bacteria. That means here you can see the effect, okay, where P32 is integrated into the bacterial genetic material. after blending. Thereafter, centrifugation. The complete protein codes and the genetic material will be centrifugated. And after centrifugation, you can clearly observe, we have learnt from biomolecules as well, that after centrifugation what happens? The dense particles you can see in the retentate at the base. The dense or molecular weight wise rich molecules are present at the base in the form of retentate, and lighter molecules are present above in the form of filtrate. This is retentate and this is filtrate. So, here if you see, when no radioactive S32 is detected inside the cells, because it is not protein code that is blended along with the genetic material of bacteria, it is the genetic material of bacteriophage that gets integrated with the genetic material of the bacteria. Got it? So, when the radioactive S32 is not present inside the cells, that you can observe after the blending, and this you can see in the supernatant. S32 is observed in the supernatant after the centrifugation, right? S35 is observed where? In the supernatant after centrifugation. But here if you see, radioactive P32 is present inside the bacterial cells, because it gets the genetic material of bacteriophage is integrated into the bacteria, and you can see no radioactivity that is detected in the supernatant, because all the protein codes are light in weight, right? When compared to the genetic material, protein code is light in weight, and that is observed in the supernatant. Here you cannot see anything in the supernatant. There is no radioactivity detected in the supernatant. But here in the first phase, radioactivity is present in the supernatant, that is S35. With this, it is proved that that is the genetic material which is transforming into the bacteria, but not the protein code. This has given the unequivocal proof that DNA is the genetic material. Once you go across the complete experiment, if you are having any doubts, please put it forward. I will answer your doubts. And then if this is clear, we are now clear on the point that DNA is the genetic material which is transforming across the generations. First from transforming principle by Frederick Griffith, second by biochemical characterization which is proved by 3 scientists, that is Oswald Avery, Colin MacLeod and M.C. Carty, and third Hershey and Chase experiment from bacteriophage. Got it right? With 2 different strains of radioactive isotopes, one of the protein code that is S35 and second that is P32 which is from genetic material. Got it right? Clear till here? And then now when we have to discuss on replication, there are 3 modes of DNA replication from central dogma of molecular biology. First come first, when nuclein is present inside the cell, which is the acidic substance. The acidic substance, that is nuclein, which is identified by Frederick Mister. Nuclein which is identified by Frederick Mister. At the same time only, from principles of inheritance, Mendel has drawn out his observations. Like from monohybrid cross, dihybrid cross, he has given the principles of inheritance when at the same time nuclein is identified in the cell. Later after, the genetic material is DNA and that is transforming is proved by the 3 scientists. But then from central dogma of molecular biology which is given by Francis Crick, who has given central dogma of molecular biology. Where the information from DNA is transferred to mRNA and then to proteins. The ultimate goal of DNA is to synthesize proteins. From central dogma of molecular biology, the very first step is what? Replication. the very first step is replication. 3 modes of DNA replication is being identified. 3 modes of DNA replication. First is conservative methods of DNA replication. First, conservative method of DNA replication. Second, semi-conservative method. And third is dispersive method of DNA replication. The first one, that is conservative method of DNA replication. Here, if I have to suppose this is the parental DNA, five prime to 3 prime, 3 prime to 5 prime, anti-parallel to each other, right. So, when this is the parental DNA is as it is cloned to a daughter DNA. It is cloned to a daughter DNA like this, from 5 prime to 3 prime, 3 prime to 5 prime. Got it? So, at the end you can see one parental DNA double strand and one daughter DNA double strand. The double stranded parental DNA is giving rise to a double strand daughter DNA. Fine, right? Here both the strands are acting like parental strands. 2 strands are parental strands only. And at the end it is giving 2 daughter strands. Got it? So, here from the conservative method, what it is saying, this method of DNA replication, wherein from a double stranded DNA, double stranded daughter strands are formed. So, at the end you can see 2 double stranded parental strands, 2 daughter strands. Here both the strands are acting like parental strands only. In the semi-conservative method of DNA replication, if you consider this as the parental strand, 5 prime to 3 prime, 3 prime to 5 prime. Each strand is a parental strand, but then here you can see each strand is the parental strand. One is from 5 prime to 3 prime, second 3 prime to 5 prime. To this parental strand, daughter strand is being replicated separately. Okay. Synthesis will always be from 5 prime to 3 prime. Synthesis will always be from 5 prime to 3 prime. Got it? Here 2 parental strands, 2 strands are acting like template strands and 2 of them will have a daughter strands. And in the product, if you can see the hybrid strands with one parent and one daughter strand. In dispersive method of DNA replication, if this is the parental DNA, from 5 prime to 3 prime, and 3 prime to 5 prime, you can see the bits and pieces of the parental strand will act like templates and daughter strands are formed like this, from 5 prime to 3 prime and 3 prime to 5 prime. Same the case with the second strand. Yeah, the diagram is a bit uneven, but that's fine. If you understand the concept, that's okay. So, this is the dispersive method of DNA replication. The bits and pieces of the parental strands will have in the daughter strands or in the products, if you see, the bits and pieces of the parental strands will be, bits and pieces of daughter strands will be replicated as such. So, from the 3 modes of DNA replication, that is 3 models of DNA replication, semi-conservative method of DNA replication is considered as the most accepted model. Most accepted model of DNA replication is semi-conservative method of DNA replication, where the 2 parental strands act like templates and each strand will have a daughter strand. At the end of the products, you can see the hybrid strands. To prove that semi-conservative method is the most accepted method of DNA replication, Michelson and Stahl's experiment is present. Okay. Michelson and Stahl has performed an experiment by taking the model organism, that is Escherichia coli. Okay, so the third experiment, so the fourth experiment that you are going to learn from this chapter is Michelson and Stahl's experiment. Michelson and Stahl's experiment. Michelson and Stahl's experiment. So, here if you see the parental DNA strand, this is from Escherichia coli. What is the model organism here? E. coli. Remember, the generation time, which means the replication time, the DNA replication time of E. coli is 20 minutes. One DNA will get completely replicated into 2 daughter strands in 20 minutes. So, the generation time here is 20 minutes. Here we go. First, E. coli is cultured. E. coli. This is cultured with a heavy nitrogen isotope, that is N15. Okay, N15. N15 isotope, this is cultured for some generations, for many generations you can say, many generations, wherein N15 isotope got integrated into the DNA of the E. coli. So, the first you are taking N15 DNA, got it? So, initially what is done, you are clear with the Michelson and Stahl's experiment and the model organism, that is E. coli, whose generation time is 20 minutes. In the first phase of the experiment, what is done, the E. coli is cultured for many generations by taking heavy isotope of nitrogen, wherein this heavy isotope got integrated into the genetic material of E. coli. And in the first phase, what is there, N15 DNA of E. coli is present. Okay. N15 DNA of E. coli is present. After 20 minutes of incubation in N14 medium, this is N15 DNA. After 20 minutes of incubation of N15 medium, of N15 DNA in N14 medium, you can see hybrids, that is one strand N15 and one strand N14. Here you can see N15, N14. Here also you can see N15 and N14. How did you identify this? By conducting centrifugation. After generation time, the culture medium is centrifugated by taking a centrifugation gradient, that is cesium chloride gradient. With this cesium chloride gradient, when the entire medium is centrifugated, you can see only one band, that is N14, N15. Got it? First come first, E. coli is cultured with heavy isotope for many generations, where N15 got integrated into the genetic material of E. coli, and then only for one generation this is being cultured with N14 medium, that is normal nitrogen, and then in the first generation you can see N15, N14. How did you identify this? By centrifugating this culture medium with cesium chloride gradient. Okay. wherein you got only single bond, that is N14, N15. And then again, when one generation time is considered, that is 20 minutes, you will get the second generation. Now here each strand will act as parental strand, right, according to semi-conservative method of DNA replication. Each strand is the parental strand. Now you tell me how many parental strands we have from generation 1? 4 parental strands, that is N15, next N14, again N15 and N14, right. All this will act as what? Parental strands itself, and this is cultured where? In N14 culture medium only again, right. This is cultured in N14 culture medium only. After culturing this, you will get, after culturing in N14 medium, to all the parental strands, N14 itself is added. N14, N14 and N14. Post this, when you centrifugate it with centrifugation medium, that is cesium chloride, when you centrifugate it, what you will get? 2 strands, one at the base and one at the top. Got it? That means you get 2 hybrid strands, 2 hybrid strands, and 2 N14, N14 daughter strands. 2 hybrid strands and 2 daughter strands. Got it? You will get 2 hybrid and 2 daughter strands, one at the base and one at the top. Got it? So with this Michelson and Stahl's experiment, it is proved that semi-conservative method of DNA replication is the most accepted or widely accepted model with this experiment. What type of questions we will get from Michelson and Stahl's experiment? Quite a times you can see in EAP set and in NEET as well, you will get application based questions. In this experiment, in the first phase, we have taken N15 isotope for both the strands. But then, if we have to consider N14 medium itself, N14 itself in the first phase, without any heavy isotope. The application based question, one model I am telling you. This if you have to consider in the first phase, and if the question is like in the first generation, you have incubated it in N15 medium and you got the products, and in the second generation you have incubated in N14. So here after second generation, what is the percentage? What is the percentage or probability? What is the probability of light strands, hybrid and heavy strands? In simple terms, what is the probability or ratio between N14, N14, N15, N14 and heavy, that is N15, N15? What will be? Okay. So here if you observe in this case, when 2 strands are N15, N15, you will get, when it is incubated, here 2 strands are N14, N14. To this, when N15 culture medium is added in the first generation, you will get 2 hybrids itself, right? 2 hybrids itself. And these 2 strands, when they are incubated in the normal culture medium again, after generation 2, what do you get in the normal culture medium? You will get N14, N14, then N15, N14, then N14, N14, and then N15, N14. The result is same. So, N14. now it is okay. So, here at the end what will be the probability that you will get? Here you will get 2 hybrid strands and 2 light strands and 0 heavy strands. So, it will be 1:1, 1:1, that is equal proportion of light and heavy strands that you will get. Such type of questions, they can give you an incubation time of 60 minutes. Incubation time of 60 minutes means how many generations? 3 generations. They can give you an incubation time of 20 minutes, which means 1 generation. So, based on that you will get application based questions from Michelson and Stahl's experiment. Finally, the key takeaways of today's session. What did we learn today? The experiments in the previous classes of Mol Bio, we have clearly dealt on DNA, RNA structural components and the differences between them. Today we have proved, we have understood why DNA is the genetic material, what are the major portions of discoveries you have to learn from molecular basis of inheritance. First, that is transforming principle by Frederick Griffith. Second, biochemical characterization, and third is Hershey and Chase experiment. And then you have learnt on 3 models of DNA replication, post which we have looked the model or the experiment that is given by Michelson and Stahl, proving that DNA is the genetic material. DNA will replicate in semi-conservative method of DNA replication. Got it right? So, these are the key takeaways of today's session. Tomorrow we will be coming up with a new concept on Mol Bio chapter again. Thank you.